patience. Okay, you can hear me now. That's better. All right. Let me just briefly introduce myself and some of the problems I've worked on. I'm an experimentalist who likes to work on problems in cell motility. And I really got fascinated by these questions when I was a PhD student, where I worked on swimming bacteria as active fluids and looking at the effect of adding polymers and adding complexity into the solution and how that affected cell behavior. And I was fascinated by this interesting connection, right, this interdisciplinary challenge of how cell behavior can affect the mechanical environment, and the mechanical environment can affect the cell behavior, right? So there's a coupling there. If you change fluid viscosity or fluid viscoelasticity, a cell may change its gait or force production or possibly gene expression in some way that can also remodel and affect the environment around it by degrading it or inducing shear thinning effects. All right, and so I've started my own group now at Syracuse University, and we're working on two main problem areas. We're looking at collective cell behavior and also single cell behavior and bacteria colonies, which I'll tell you about today. We also are studying animal cell migration, particularly fibroblasts, and how they relate to the extracellular matrix. And this was work that stems from my postdoctoral work with Paul Jamney. And although it's not the focus of my talk, I'll just give a highlight that we have a focus on the third cytoskeletal polymer network. We heard about actin and microtubules during this conference. We work on vimentin intermediate filament proteins, which we've shown are really important to the mechanical structure of the cell and protecting the nucleus from damage, and regulates things like cell size and traction forces. And we've also started recapitulating these networks in vitro. And I'm starting to do some interesting rheology on them themselves. They have these interesting nonlinear shear strain stiffening and compression stiffening behavior that interacts also right with the other cytoskeletal networks and composite. All right, and also as a means of introduction, I'd like to highlight that I think of bacteria as very complex systems that actually are able to mimic some of the more highly intelligent behaviors of our eukaryotic systems. And so one example is they can undergo forms of development. And in this case, this is a soil bacteria, Myxococcus xanthus, which I'll come back to at the end of my talk, which when starved, they aggregate into these what's called fruiting bodies that has spores that can detach and then go hopefully to a better environmental space. But there's a system of genetic programming that happens, and it's considered a form of development. Okay. All right, and so I'm going to start with this problem, looking at how colonies are interacting with their surface, right? That interplay of how the physical properties change the colony behavior. And this is a standard experiment where you plate bacteria. This is Serratia marcescens. For all intents and purposes, you can think of them like E. coli, or from the physicist's perspective, you can. And on soft agar, you see the colonies spreading out much more rapidly compared to stiff agar, where they're spreading out much slower. And so this is, a clear demonstration that the mechanical properties of the environment, like we saw yesterday, are impacting how the colony moves. But I like to think about analyzing bacteria dynamics on agar, like studying humans swimming or running across cornstarch. Okay, agar, like cornstarch, is a non-Newtonian material with complex material properties. It's a viscoelastic material, meaning it has viscous dissipation, also elastic spring-like properties. And the point is, because of the complexity, one, it's hard to understand, and two, the phenotype of the organism may be doing things it might not normally do if it was walking on something mechanically much more simpler. All right, so the two problems I'm going to tell you about today are building some tools to quantitatively look at how colonies move and generate force on platforms we can understand much more easily in a mechanical sense. And then also this question I like to call mapping the bacteria phenome, which I think presents some unique challenges. And by phenome, I just mean all the phase spaces, all the things that this collective bacterial motility can do. What can we make them do? That is the phenome for the collection of phenotypes. All right. So our approach to designing these substrates is really taking a cue from some of our animal cell study work, where we're using polyacrylamide gels, which have been now used for a couple decades in animal cell systems, where an animal cell will change its structure and its phenotype on a soft gel compared to a stiff gel. And we have to do a few tricks, but you can get bacteria to grow in them too. And so these gels are advantageous in many aspects, and in one way they have very well controlled and defined mechanical properties that we can tune. So they're very linearly elastic. This is an oscillatory shear rheology plot as a function of shear strain. And they basically have no viscous dissipation. This is basically noise from our machine. Okay. Right, and this is in contrast to agar, which one softens at higher strain. So maybe depending on the relevant strains or frequencies of the problem, it's possible that bacteria would experience a different stiffness, but they're also experience a lot of viscous dissipation in the system. And also note, we can tune the gels and the pore size of the matrix, which is thought to be really important for how colonies grow on substrates, the pore size, which allows nutrients to diffuse out. If we can add more monomer to make the gel stiffer, we're going from about 500 pascals to 30,000 pascals, which is through a range of tissue stiffnesses. It's just a comparison. And we can also tune the stiffness by adding more crosslinker, which does not change the pore size nearly to the same extent as increasing the monomer, but has a strong effect on the stiffness of the matrix too. Okay, and so it's a relatively simple experiment. We have a hydrogel. There's an air interface here, the hydrogel filled with nutrient-rich medium. We put a colony of our bacteria here, and we watch the edge as it expands. Okay. This was work mainly due from my graduate student, Meryl Asp, who graduated in December. And our pandemic project was to come up with a semi-automated code that we now have open source that allows you to track the colony as it expands. Of course, anyone who's worked with image data knows it's very hard to get these things perfect. So that's why there's kind of a semi-automated, allows you to just identify when there's defects and then fix them. All right. So let me just re-highlight as a control what here we go, agar. What happens for colonies on soft, which is low concentrated agar compared to stiff agar. Let's see, is it playing? All right, reminder, it's faster here on the soft substrate, much faster. And so the colony expansion rate that we measure is decreasing as the agar stiffness increases. Now we do the same thing on polyacrylamide gels of different stiffnesses. If you watch, there's this, at one moment, this takes off and has a much faster expansion rate. Okay, so this is opposite what was perceived on the agar substrate. And so as a function of increasing stiffness, remember this is linear elastic, no viscous dissipation, we get an increasing expansion speed of the colony that seems to saturate and in some cases goes back down. And because I'm going to kind of gloss over this a little bit, we see this trend whether we increase the monomer, increase the stiffness by increasing the monomer, or the cross-linking context. And we see a very similar trend. I think there are some effects of pore size, we think, at these smaller stiffnesses, but this is highlighting that the stiffness of the substrate is contributing to the speed of the colony expansion. Okay, and I just want to highlight this isn't specific to Serratia marcescens. We also wanted to check whether it occurred in other bacteria. So here I have Myxococcus xanthus, that's the soil bacterium I showed you previously. Interestingly, they don't have flagella, but they have pili. Pseudomonas aeruginosa, and 2 strains of Proteus mirabilis, which are very interesting swarming species. Okay, so one of the questions we wanted to ask next was, okay, if there's this hypothesis that the cells are somehow sensing the substrate and changing their behavior, or maybe they're doing it passively, right? They can only really sense the substrate stiffness if they're able to deform it, right? Apply some displacement or force to that substrate. And so to analyze this, we wanted to embed the substrate with tracer beads where we could map the displacements on the surface right below the colony. And so here's a 2-channel imaging. We have the bright field colony edge, and here we have the fluorescent beads in the substrate. And I'm just going to play you a version of this on loop, where every other loop we have the boundary position highlighted in red. And there's kind of a pulse that's moving with the expanding edge of the colony. And this is just another example. This colony is moving from this edge down to here. And here the beads are just on the surface of the colony. So you can see that pulse moving a little more clearly. Okay, and so from there we use PIV to map the displacements over time of the colony. And you get these very complex structures that are primarily here at the surface of the expanding edge. Interestingly, in some cases, we see displacements that are ahead of the propagating wave. And there's local kind of hot spots, which have been reported before in some bacteria species. I'll note here we use a finite element model based on the elasticity of the hydrogel to estimate what the stresses are on that surface. And I'll just highlight, right, this would be a much more complicated problem on agar because, well, we haven't tried to solve it yet, but it's not such a one-to-one relation between the displacement and the surface displacements in that case. Okay. Now, those were kind of if you watch the video at a high time resolution, but if I also like to watch things in what I call fast time and slow time. You see very different things, and so if you watch the system over many hours, say 15 hours, there's a net contractile motion of the substrate from the colony edge towards the center that builds up over time, all right? And actually this had been, something similar had been seen previously, actually from Alex Prasad's group, where the colony is actually able to deform the substrate to some degree and pull it in as it generates stress over these long times. Okay, so now that we had this system and we could tune the substrate properties very well, we wanted to ask, how does the force generation of the colony, which we kind of read out from the surface displacement maps, depend on the substrate stiffness? Is the colony adapting in some way to these physical cues? And so what we have is in our window, we can map displacements of the particles over time, and we calculate an average stress over time, and so it's increasing over time. And what we found was that on these soft gels, which were around 500 pascals, we had up to 100 pascals, but compared to a stiff substrate at 5 kPa, it was up to 600. So a greater increase in the force generated on these stiffer substrates, which was not clear a priori. All right. Now, I've given this talk once before in the States, and usually I tell the physicists to close their eyes at this slide because the biological complexity can sometimes be daunting to theorists. But this is an interdisciplinary challenge workshop, so we can embrace it. I know, I'm fully aware that there's many factors here that could be contributing to how the colony is changing their behavior on the different substrates, which is ultimately what we want to know. And we're starting to explore some of these paths. One thing I'll note is that, and I often get asked if there's a difference in the wetting properties of the gel, because the surface wetting properties could aid the expansion, but using some measurements, we've actually found that our stiffer substrates, where they spread out faster, actually have less wetting, and so that goes against the hypothesis that it's the surface wetting properties of the substrate itself. And because of the complex patterns we saw at the expanding edge of the substrate, we've become really drawn to some of the models of biofilm colony growth based on osmotic spreading. And so these models have really started in the Brenner group, and now other groups have looked at this, where colony expansion is mediated by the ability of the colony to take up fluid by its substrate, and the colony actually has this kind of active pumping mechanism in some way where they are excreting these EPS molecules or other molecules that can act as osmolytes, which pumps fluid from the substrate into the colony, allowing the colony to take up more mass and to expand. One of the lines of inquiry we think is happening here is that because of the poroelasticity of the substrate, as fluid moves from the substrate into the colony, the flows induce deformations in the substrate, and that's what we're picking up at the edge as it expands. And it's all very hand-wavy at this point, but perhaps a stiffer substrate is better able to transmit those stresses that allow the fluid to come out and better allow the biofilm to expand. I think this would be a great project for finite element modeling, but we haven't gotten anyone to take the bait yet on that problem. Okay, so what's happening on these agar substrates then? Right, I often get asked this question: why are they different? Why are they slower? And so one thing we might think is important here is the viscoelasticity of the substrate, and I'll show you some evidence of that. By making a viscoelastic substrate, that we can tune the viscoelasticity and see how the colony responds. And so there's different ways you can make a viscoelastic gel that's tunable. One of the ways in the literature is by adding crosslinks in the gel that are dynamic and turn on and off. This, though, turns into a substrate that's viscoplastic, so it won't actually return back to the state it was if it's sheared, depending on the timescale deformations. And so instead, we've turned to an alternative method, which is we take these linear viscoelastic, or we prepare these long linear chains of polyacrylamide that act as a viscous dissipation unit, and we polymerize our cross-link network around these chains, right? And these chains are very long. They have to be very long. If you can pipette them easily, we know it's not going to work. And they get trapped in the meshwork of our cross-link gels, and it forms a very viscoelastic material that is truly elastic at long time scales. It will recover. And so we can get the same range of stiffness. Here we're going to just hold at 5 kPa shear stiffness. And when we introduce the viscoelasticity, we can get about 10% of our G prime. So in this case, 500 pascals G double prime, which I'm going to show you actually has a strong effect. So it's in an experimental range, fine, but I'll also say it's actually quite challenging to get it higher than this with this approach. Okay, so, and what we see here, this is mostly a snapshot because this is ongoing work, is that on the viscoelastic gels, typically we get a much slower expansion. And so it could be that viscous dissipation in the substrate is impairing the colony expansion rates. And so, and this is clearly seen after overnight with your iPhone, you can see that the colony is much smaller on these viscoelastic substrates compared to our elastic ones. I thought I took this out. Maybe I didn't. I'll just highlight too. We've discovered on our very soft substrates that we also see this tearing and ripping on the colony. This was perhaps a little akin to what we saw yesterday, but the boundary conditions are different. It's on a hydrogel surface, and it's something we're interested in pursuing a bit more. Okay. In the last 5 minutes I have, I'm just going to map out a problem that we've been working on that I think might be of interest to this group, and I'll motivate it. I came to a conference a year or two at Aachen and was inspired by this kind of quote or story that, the 19th century was about mapping the world. In the 20th century, we were mapping the atom, and now we're mapping the cell, so kind of a call for mapping everything we can about the cell. And in this way, we are mapping the collective motion of bacteria colonies, and here in this problem, I'm going to be talking about this developmental program of fruiting body formation where cells, they're on a surface and they aggregate together and they form actually these three-dimensional domes. And since joining Syracuse University, I have a colleague in biology, Roy Welsh, who's really an expert on mixo and their collective pattern formation. And what he's done is he's also fascinated by all the things mixo can do. And he's generated this huge library of different knockout, single gene knockouts. And you can see you have very different structures that form. In some cases, they don't form at all, although it is fairly robust. And trying to figure out all the different states they can form, how they form them. This is important to being able to design programmable matter, etc. But one of the challenges is, if we want to start thinking about different gene programs and we want to start thinking about different experimental conditions, that can change their behavior, you quickly get to a huge phase space, So challenge is, what parameters do you explore? How can we get high throughput data? Here's just a few samples of colonies that do kind of interesting things that are much different. I don't know what the genes are. There's some random gene. Okay. So to scale up this problem, we've been taking this approach where we're using, relatively cheaply built microscopes. This is a 3D printed microscope with a 4x lens and a little stage to control the temperature and whatnot of the system. And we built a whole array of them. Okay, so we have 200 of these 3D printed microscopes. Each one is an independent experiment with temperature control and a camera. And what we've helped wire all of these up to a central computing station that can collect all the data, and also found a way to track these fruiting bodies over time. And so here we have them colored green and orange for two different populations. The green ones we call persisters because they stay. The orange ones you'll actually notice end up disappearing by the end of the movie. This is a 24-hour movie. And so we have temporal resolution, I think, to a much better degree than has been done before for this fruiting body problem, where this is each fruiting body. Blue marks the time that they're their biggest size. And we're gathering this wealth of data. Okay, and a few years ago, there was a paper saying that this process could be modeled as a motility-induced phase separation process, where each bacteria underwent a persistent the long conference is hitting me now. Persistent self-driven walk, okay, with some diffusion and some length scale of the walk. And you could get either nucleation and growth or sphenoidal decomposition, phase separation of the particles into these aggregates that followed some scaling. Here alpha equals 0.3. And I don't have an answer to this, but we actually find that, especially for different mutant classes, or actually for our wild type, we can't actually recapture that phase scaling. This is a problem that we're actively recruiting people to our group or collaborators for right now, to help us understand all these different mutant classes, how they assemble, how they do things different from motility-induced phase separation. What does that mean about the physics of emergent systems? Okay, so yeah, we're working on building hydrogel systems to look at fourth generation of bacteria colonies and trying to map out different phase behaviors and Myxococcus xanthus fruiting body formation. And yeah, with that, I forgot to say thank you to the organizers for inviting me to this wonderful conference in wonderful city. And I'll take any questions. Thank you.
Thank you very much, Alison, for a beautiful talk. Any questions? Yeah.
So you mentioned that agar has shear thinning, right? Or like a nonlinear elastic property, right? And I was wondering if you know what that does to the motility.
Let me see. I understand the question. The question is,
So the
how
agar
does she-
gel
Yeah.
has a nonlinear elastic property, right? It has shear thinning, is that?
The hydrogels we make don't have any
Right.
shear thinning effects, but agar in itself has some.
And
yes.
do you understand what that does?
No, we have not. Yeah, we wanted to understand the simple case first.
Yeah, all right, thank
But
you.
yeah.
More questions there?
Do your colonies secrete a lot of surfactants? Because I was just wondering if that could have an effect too, because it would lead to swelling, like when there's surfactants, and maybe it would go with your picture of local swelling, and then like maybe it would help like the process too, or maybe it's not critical in your colonies. I have no idea.
They are, yes, excreting surfactants. We have not characterized it under different substrates. We could add surfactants to the surface, see how that changes things. It's definitely, yeah, something we should look at. Yeah.
Have you characterized the way the mutations affect the motion of single bacteria for the MIPS problem? Or is it you really look at the mesoscopic scalings, but we don't know necessarily how it affects, say, the persistence length of the random walk?
Yes, that's a good question, and something also that we want to do. Because, right, if you just knew the difference in speed or persistence length, maybe the MIPS model would give you the phenotype we're seeing. I have various reasons for thinking it's not going to capture everything we see, because we have some very strange phenotypes, but that's definitely a direction we would like to go, yeah, to check how far it fits.
We have time for one more question, and we can continue the discussion during the coffee break.
Thank you for a really nice talk. I was wondering if there's any systematic, if you mentioned like the growth of the colony is faster at the beginning, then slower after. Is there any systematic trend, or is this just kind of random? And then you just pick up an average speed, I guess, of spreading?
Oh, yes. Yeah, okay, I have the data here. These are on the hydrogels, although I think something similar happens on agar, although the trend with the substrate might be different. It is very time dependent, also like we saw yesterday. And so typically there could be a lag time for reasons I don't think people quite understand why. And then it peaks which is thought of to be because of some nutrient depletion and then slows down. And so there's a lot of temporal data here that we're also digging into. But for the purposes, we've looked near this initial expansion rate where there should be lots of nutrient availability. Yeah.
Okay, let's say thank you again to Alison.